All right. So just to remind you, basically the first part of the challenge, and the second part we didn't get into, but first part of the challenge was just trying to predict a network based on perturbations of the different nodes of the network using drugs. The second part of the challenge that we didn't discuss, but would be once you have found the network, what we ask you is, can you try to predict time courses of other types of modifications that can be done in this network, perturbations that can be done in this network, and um, uh, to see if you are also good at predicting time courses, which is something we're also interested in Dream. So not only having the network, but then once you have your network, how good are you at predicting time courses? I mean, you could think that this is very correlated, but you would be surprised that actually it's not completely correlated. I mean, you definitely have to optimize your algorithms also for doing this. Okay, so just the basic idea of the scoring of this, and there are different ways, and we'll discuss a little bit this, is basically using area under the ROC curve. So receiver operator characteristic curve, where you plot false positives versus true positives, and you basically measure the area under this. And in this case, just very briefly, what you do is just basically measure distance between each of the time points, and that's what gives you your distance to the gold standard, right? So the predicted is the one in kind of pinkish, violet, and the blue is the measure. All right. So again, the first challenge, you have node values over time with that. And with a different, either DMSO or different drugs that act, for example, on mTOR, and so you want to see how the drugs affect further downstream nodes of the network. So the true causal network underlying the variables in the in silico data was obtained from the data generating nonlinear ODE model. So we basically, it's an in silico challenge, but we want it to be as close as possible to the reality. And so we used a pathway, a biological pathway. Basically ERB signaling through GRB, RAS, here the MEC cascade, and the PI3 kinase cascade, and this is basically this complex network that we wanted to make into a more tractable. So this is basically the gold standard network that we used to calculate the difference between your predictions and the network. So it's just based on a real biological network that was transformed into another network. So I don't know how similar it looks to your network already. I mean, you can already see the image, but we're gonna dig into that.
We didn't get that edge.
Which one? You remember it was there was one that was very centralized. Probably is this one, right? Is the receptor. So one thing is, as you saw with your data, we didn't tell participants that, what the network was, of course. So they didn't have any data on anything related to the biology underlying the network they had to predict. It was really only data-centered. Actually, the participants that did on the other challenge were told what was measured. So that was one of the differences between the in silico and the in vitro. Okay, so basically you guys submitted a network matrix, as we saw, that has confidence values for each of the edges. So what do we do with threshold? We choose a threshold and we discretize a network matrix between ones and zeros, and then we compare that to the gold standard. And by varying the threshold, we obtain different false positives and true positives. And we compare these by comparing it to the gold standard. So basically, by varying the threshold, we obtain a different each time different value of the number of false positives and the number of true positives. And so we obtain here an area under the receiving operator characteristic curve. And by just calculating this area, we obtain a score of how well you did. And I don't know if you're familiar with this kind of curves. Are you? Area under receiver operator characteristic. But basically, the idea is that you want to balance, how many edges you got correct to how many edges you missed. Right? That's basically the idea, because you can have a lot of true positives, but basically miss a lot of the edges. And by having more false positives, you can balance the number of true positives. So that's the idea of this pretty standard and common approach of trying to score a problem where you can, by thresholding, make it into a decision of whether it's true positive or false positive.
So one thing that we always think is whether there are other metrics that are good for this, and in this case, definitely one of the metrics that we could have used is area under precision recall.
Yes, right? Area under precision recall. So I'll discuss and compare that a little bit more. So we're just discussing how we scored this challenge, and maybe just briefly, and the fact that we used the real network that was converted into a silicon network. Of course, you guys didn't know what the network was, but it was really trying to be as close to the real biology. And basically, for scoring, we use area under the receiver operator characteristic, which basically is plotting false positive versus true positives by comparing the network obtained with a certain threshold for the confidence to the real network, and just basically counting how many false positives and true positives you have. And so when you plot that for different thresholds, you obtain this curve, and then the area under the curve gives you a measurement of how well you're doing compared to how many nodes you're missing. That's basically the idea, right? You want to know how precise you are, and compare that to how many nodes you're missing. Yes?
So once you had your in silico model, what else did you do to the data? You added some noise. You looked at the real data set to have a model of the noise to apply to the in silico model?
You mean for the...
So how to generate in silico data set?
So yeah, so you calculate it from the model that Pablo said, and as you said, you use a noise model which has both like a more Gaussian term plus some offset term, more or less based on what you think is the noise of the data.
Was it the Langevin stochastic differential equation, or was just adding what would be measurement noise?
Yeah, it would be other ways to do it.
This is for the time course predictions, you mean, or? Yes.
Well, originally how the data was generated,
sure.
Right? It was not the ODE.
Sure.
Maybe we should have mentioned this. It's not the ODE values that added noise.
Yes, sure.
The data that they got to participate.
Yes.
And the noise, you looked at the noise from the experimental data set?
Yes.
Yes.
I'm sorry to join late. Can you just quickly tell me the input data that was used to create that the contestants are using to create that network matrix?
Yes, sorry. So basically the participants obtain what they got is 5 perturbations, I think.
Well, combined, then large number of conditions. It's, two ligands and 3 drugs are put in the cells, or in the model in this case, in different combinations. And for each of those combinations, we generated time series data of 20 different proteins.
And this was mimicking real data that also was offered in another sub-challenge. Also in the real sub-challenge, in the real experimental data sub-challenge, we didn't know what the gold standard was. So we had to use a surrogate gold standard. In this one, the same was simulated, the same type of perturbations, inhibitors, different stimuli, and their domination, we knew. So we were expecting to see whether the best performing in one would be also best performing in the other. so I think we discussed.
Yes, yes. And basically, I mean, one of the tricks we did is that 3 of the proteins were not part of the network. But basically, if you count, here are the 20 nodes, and the idea is that we were using, again, different ligands here, as Julio said, 2 different ligands, and then 3 different drugs that would hit on different, differently on. I mean, would they hit on different nodes? Yeah, I mean, they would affect, several variables. So that perturbation data was used by the participants to try to reconstruct the network.
And the measurements are phosphoproteomic?
Yeah.
Yes. Using
RPPA.
RPPA arrays. Okay. So basically, right. So what we get is the different with a different score in the different contexts, we get different OROC ranks, and then we get a mean rank and a final rank for each of the submissions, and that gives you your basically how well you fared. So this is just a brief example from the actual challenge, just to go very brief to see all the different kind of approaches that are shown here. So there were 65 submissions in the challenge, and each of the colors is a different method. So you see that basically the methods were not really how well the methods fared is not really clustered to the different kinds of methods, and you see that the best method had a area under the receiver characteristic OROC of 0.8. These are just the first. So basically, the first I think 13 submissions were statistically significant, better than I think you use random for as a comparison, right, Julio? Just random submissions. Yes.
So we just saw that 65.
Yes.
That would be in the top 10.
Yes. Yes. Well, but you had 65?
No, I didn't.
No, right.
That was not right. That would have been in the top 10.
That would have been the top 10.
It's not the top.
Actually, we used one of the ones that were here.
Yeah.
Why didn't you choose the top? Just because.
Yeah. Actually, because they didn't make it public, so I didn't have access to it.
Oh, I see.
Okay, so this is just to show you, one of the things that we do is we resample some of the nodes in order to see how robust the score is, and to be sure that it's not only related to how we chose, the data or very specific to an algorithm, but that it's pretty robust. And as you can see clearly, the best performer here, the score didn't vary a lot, and could still be separated from the second
right. And
best
this
performer.
Is, in general, something that we try to do, right? We had a choice of. We always have a choice when we run challenges of saying, okay, the winner. I don't like to call it winner, but we call them best performers. Is the euphemism. So the best performer is the one who got 0.65. The second one got 0.64. Therefore, the best performer is really the best performer, or is it statistical? No, that actually, if we had changed a little bit the composition of the test set, the results would have been swapped. So we always do these kinds of analyses to show the robustness of the scoring, and we don't call best performer the best performer. If there is a tie, we usually call all the ones that are more or less at the same level after reshuffling best performance. Sometimes we have 3, 4.
Yeah, and we declare them, all best performers. Maybe. If we cannot differentiate, we cannot differentiate. There was one challenge that we got. Everybody was best performer.
It was a flow cytometry challenge, and we wanted to see separate AML patients from non-AML patients. We did jointly with flow lab, and everybody was separate because it was a very easy challenge.
Okay, so again, this is just now clustered by the types of approaches. As again, you can see, you notice the same thing that Gustavo talked about before. That it's not because you use one method that you're gonna be better. It's really how you apply it. So almost. Although I mean, sometimes there's a tendency for a method being better than others, but overall, you see that. I mean, these guys seem to like not fare very well, basically.
Right. You see, Bayesian network, in Dream at least, never got in blind testing a good performance. Neither regulatory network or so. Don't use it.
All right. So just to make the discussion on scoring, to just point the fact that choosing scoring seems to be kind of trivial, but actually there's a lot of discussions around it because you could measure in different ways something, right? And so just to show you that an alternative way of measuring the same thing would be to use the area on the precision recall curve. So the precision is something a little bit different than true positive and false positive. Is basically how many true positives you have for the number of edges that you choose. And the recall is the number of true positives you have over the total of edges, positive edges that exist. And so you can plot, plot a curve of precision versus recall. In a sense, it's a different way of measuring the same thing that I was telling you, true positive versus false positives. And basically, you choose your index, so basically how many nodes you're going to choose. This is just an example. So for example, and in general, you align them by the confidence score. So you align all the nodes by confidence score, and then you go down the confidence score one by one, and you calculate the precision and the recall. So for example, in this case, he got it positively, so his precision is 1, and there's 10 nodes, so the recall is 1 in 10, right? So if you go to the second one, now he got it incorrectly, so now his precision is 0.5, so 1 out of 2. His recall is still the same because it's still 1 out of 10. So you continue, right? And you see that the curve basically varies and is measuring a little bit, again, the same thing of how well the participant is doing. So, right, so why did we choose one or the other? Well, at the end, the answer isn't always easy of why we choose one way of scoring or the other, but we can have a post hoc answer, which is let's try to see how well correlated are the ranks of the teams by using one measure, a UPR, which is the one the second method I was showing you, or the first method, which is OROC. And as you see, luckily, if you take only the in silico data of participants, it's pretty well correlated. So it means that although there are a couple of outliers, both methods seem to basically be telling you the same thing. And if you take both the real data and the in silico data, it gets the correlations it's even better. So just to show you, these are the post hoc ways where we sometimes just check to brainstorm that we selected correctly the scoring. And it has happened in challenges that we had to change the way of scoring because of realizing that, and in general with feedback from participants that showed some clear reasons for changing the scoring in the challenge. So even with that, we're pretty open. I mean, we prefer not to have to do that in the middle of a challenge, but we definitely assess that. Okay, just the second challenge. You didn't really work on this, but just to show you, the kind of things that can be obtained. So this is a time series of, the different proteins in different situations. So this is the best performer of this challenge. You see here some of the traces, and you compare this to the gold standard that's here. So you see that in a few cases he seemed to have it okay, but for example for this green and red that Gustavo cannot differentiate because he's blind to colors. So I love to put them in this color so that he doesn't see the difference. But you can see that they didn't do so well. So again, the score, and there are different ways of calculating this also, but the score chosen here was basically just the root mean square error. So basically the predicted value minus the simulated value squared, and so you add all that, you make the mean, and then you take the root of that. So basically you're just calculating a distance between the two lines as I showed you. And yeah, of course, and we divide by the error model that we had that Julio was discussing, which basically was an error model that had a baseline noise plus a noise that was proportional to the value of the protein, right? So the higher expression, the more noise you have.
I think you have a square too many. Because you have sigma square B plus sigma square S square.
The pk square, okay, right.
Ah, the p's.
Yeah, the p squared, right?
P is yeah, that is correct.
Yeah.
I thought that the p was an argument of the sigma.
Yeah. Okay, and so again, if you compare again one by one instead of having a whole image, you see again that in some cases the best performer did pretty well, in other less, but still follow the shape. But in some cases it really, were cases that had a little bit more dynamics. It was very hard to predict. So there's really not a one-on-one relationship between how good your model is and how well you're going to predict all proteins. It really depends on what protein you're looking at, and you have to find ways of optimizing that, fine-tuning your model to get better at predicting these proteins. So again, a couple of more examples. But you see, reliably, when the protein is changing a lot, it's difficult. It starts to get difficult to predict. Okay, so that's it for the scoring part. Again, these are just the people that worked in this challenge as Julio.
So let's talk about the wisdom of crowds. And so this is something. This is something a machine learning people, have been using and have realized, a long time ago. But I think that the way in which we are using it in the context of these challenges, I think, gives a little perspective to how you can use it, because let's try to compare different ways of doing things. But for example, the ensemble learners that machine learning people used to use, now this is changing, is more like in AdaBoost, different ways of looking at the same data, but always with the same type of algorithm, the same as random forest, the same as Bayes networks. Anyway, so this wisdom of crowds is a nice and fancy way, combination of words that has a nice ring invented by James Surowiecki, who wrote a book called The Wisdom of Crowds, inspiring another book that was written about 100 years before called The Madness of Crowds, in which the author of The Madness of Crowds were saying how crazy people are that collectively they can do a lot of destruction. And the other book that that this guy wrote, James Surowiecki, was rather say, oftentimes, if you aggregate the collective wisdom, you get a better answer than otherwise. And we saw that that's the case. Let's see this nice story of Francis Galton. You know this story? This is the ox. Some people don't know it, even though it's pretty well known among some quarters. So this guy, Francis Galton, is one of the late 1800, early 1900 geniuses of statistics. If I am not mistaken, this is the guy who named the standard deviation. I may be wrong about this, but that level of depth, and we use a lot of techniques that this guy did. He was also the father of eugenics, which is not a nice thing. But he wanted to show that democracy is wrong. And in order to show it, he tried to do an experiment. He was at least very scientific in his thinking. So he said he went to a county fair in Plymouth in England and used the predictions of different people that had to predict the weight of an ox. They had to say, what is the weight of this ox? And so he says, I will show that people don't have a clue by looking at how bad their predictions are. And so he computed the mean of all these things. It was 800 cards, so there were no calculators. So he worked hard for this. And it turned out the real weight was 1 pound out of this. So he concluded that, oh, again, I had it twice. He concluded that this result is, I think, more creditable to the trustworthiness of a democratic judgment than might have been expected. Because he expected that the result was going to be a disaster. And so how can you trust a crowd to choose their government if the crowds are a disaster at estimating? And here he says, maybe I was a little wrong. But this is one of the examples in which you use the balancing. Some people overestimate, some people underestimate, and that's the error. But the signal is there, and it turns out that that seems to be a very good strategy, and we will see it in other cases. When I was doing Dream 2, and we had finalized some of the challenges, I didn't know about Francis Galton, I didn't know about James Surovetsky. I just thought, maybe I aggregate these 3 predictions, maybe I get better than the best. So, and this is what I wrote in the paper that reported this. It is very interesting to note that the aggregate predictions of these 3 teams perform much better than even the best of them. I was very surprised to see this. Clearly, this shows that the intelligence behind the 3 methods can be easily aggregated to produce an integrative approach that is better than any method in isolation. This calls for a reverse engineering by consensus approach that has not yet been explored by the community. So that's how I felt at that moment. And I started to dig a little deeper. And with my colleagues in other challenges, we started to see what was at the root of this. By the way, this is, I think these were the 3 teams that I aggregated, this, and this, when I did that. And then this was the boost, aggregating these 3, come here. Then later on I aggregated all of the teams, and you can see really that the aggregation is not working so well in this range. This is the area under the precision recall curve of a particular data set. And this is the area under the rock. Okay. Let's think a little bit more about what we had said before. Remember that we had said this is what we got in the NCI Dream Drug Sensitivity Prediction Challenge. This scoring that we invented, the weighted probabilistic concordance index, had given us this, and we aggregated in order, and we said this must not be correct because we are aggregating in an order that usually we wouldn't know. So what we did in another challenge was the following. Suppose that you have 2 sets, and these 2 sets basically are 2 test sets, or 2 pieces of the same test set, which is what we did. And so we had the predictions of the whole set. So we separated a chunk of the test set and said, let's order according to these guys, and then aggregate the predictions on this set in the order of these guys. So we are not aggregating the predictions that we know how they score. We are aggregating the predictions as they score in another test set, and we then aggregate in a new test set, as if you are learning the priors. So if you aggregate and if you do that 1,000 times so that you have 1,000 splits, you don't have monotonically decreasing function, because sometimes one is better than the other. So this comes to what we were saying before about the robustness. But you know, even if you integrate from the test set that you didn't choose to order, you still get the boost. One thing that we never published, but is very interesting, is if you do this, we can discuss this if we have time later. If you do this with random data, actually, if you produce random data and you for some reason say this is a little bit predictive, you can aggregate random data and get a reasonably good result if you make the random data a little bit better than random. But what I want to say is that if you do the same thing as I'm doing here for random data without any other thing, and you order from best to worst, and the best is going to be better than the best, than the random, because it's a distribution around the mean. So the best in the random will be a little bit better. So you can still aggregate, but if you do it in two sets, this one is going to be worse and worse and worse and worse than this, for reasons that we can discuss at some other moment. But it's like there is no free lunch. So if one part of the random is correct, the other part has to be wrong, and so you aggregate wrongness. So, but the fact that here you are aggregating and you're getting better and better has to do with the fact that there is really predictive power.
So you always get an optimum of the number of aggregation when with the pattern, as the number of people estimating the ox rate, you get a better precision.
Right. This is a little. So what you're saying is that how do we know the number that we have to aggregate that is optimal?
And the fact that with wisdom of the pattern in terms of estimating the ox rate, I suspect that you get a better and better
Yes.
precision.
Yes. Yes. But there is a caveat to that. You see here, these are all better than random. You see, because random is 0.5, all better than random. But therefore, as far as I aggregate better than random, we are okay. But when we are starting to aggregate worse than random, we go worse than, we really are damaging our predictions. So if you add all, there was a follow-up to the Nature paper by Galton, in which someone was saying, hey, but all these people are experts because their livelihood in this county depended on estimating the weight of their cattle because they had to sell it. And so it is true that somehow you have to have some predictive power in what you have. If you aggregate an infinite number of things that start to be random or worse than random eventually, then you don't get anything. And the shrinkage of variance making better and better would not be so. We will see it a little bit later. This is another example of what we were, I think, discussing before, Stefan, which you were saying, what if we make a community of one? These are all the predictions of scores of survival, concordance index, by the unit, individual teams. Teams could predict more than once. That's why there are so many. Now, aggregate 5 and take at random 5 from here. You get the median that is better than the median of the individual. Aggregate 10 and you get the median that is better than the median of the 5. And in the end, you aggregate 50 and you see the variance has become much, much smaller at the maximum of possible predictability. So we discussed this before. We ask ourselves, can you be a little bit more intelligent at aggregate? How do we aggregate, by the way? We didn't say how we aggregate. If you have data of the sort of the edges, for example, in which we say we order according to the edges for which I have the most confidence. So first goes the one that I'm sure goes there. Second, the one that I'm second sure that goes there, and so on. But my order is not going to be the same as Julio's order or Vincent's order. So an edge in the aggregate is going to have the rank of the average ranks predicted. So for example, if there is an edge between antibody A and antibody 1, say, I gave it the first position, I was very sure. Stefan gave it the 10th. And Jaume gave 5. So the number will be 10 plus 1 plus 5 divided by 3. That's the way we are aggregating, what is called border aggregation or average rank aggregation. And but what if we use weighted and unweighted voting? So here it is the 3 compendia based on rank average, confidence average, weighted rank average, and weighted confidence average. In a nutshell, without going too much into it, it doesn't make a lot of difference. Okay, it doesn't. We discussed that. This is interesting as well. Again, the network inference challenge, Dream 5. Suppose that you aggregate the initial set of all the best teams. If you aggregate, the aggregate gives you this boost. Now start to aggregate the worst methods. They were close to random, but not worse than random. So you don't degrade your predictions too much. You are around the best. So you don't lose by aggregating teams if they are not systematically worse than random. Okay? If they are systematically worse than random, you will lose. You will lose accuracy. Now, suppose that you aggregate, the initial set is the 25 worse methods. You aggregate them by average rank, and you get this boost. Now you start to aggregate the best from there. This is the very best, second best, third best, fourth best, fifth best. You get a boost as you aggregate. So the aggregation gives you a boost if you are aggregating good methods, and if you are aggregating bad methods, it doesn't penalize too much what you have, especially if you have a sufficient number here. Yeah, it's the same scale. This is 40 minus log of the aggregate p-value is 40, and it's 40 here.
So the p-value
is the multiplication of the p-values for the 3 networks. Okay, so it's if you had 10 to the minus 12 or something. Yeah.
So between the second and the third of the best models, there is no increase. So was it the case that those 2 models were very similar to each other?
Yeah, let's discuss that right now. It's very interesting. So we did a principal component clustering of the predictions, and these are all. And we didn't cluster based on method, but it turned out that the clusters was enriching methods. For example, this is regression. This is clustered together correlation and mutual information, Bayesian, and here a bunch of others. So we ask ourselves, is it better to aggregate? Is it indistinct if you aggregate from different classes or from the same class? Which is more or less what you are asking. I will show later that it's better to aggregate from different classes, but this shows it. This is again the unique teams. If you now aggregate 3 that is within clusters, you take 3 within cluster, 3 within clusters, this is the median. If you take 3 from different clusters, this is the new median. So aggregating things that are a little bit different gives you a better result. And why? So here is how we said we are aggregated. So team 1 put item A number 1, item A team 2 put it at number 7, and item A team 3 put it at number 2, right? Think of it as suppose there is, and this is a big if, this is not like this, but let's assume this in order to have a hypothesis under which we can show the wisdom of crowds working. Assume that the true positives follow a distribution, and the probability of the good items to be first in rank is like this. So the probability of putting, this is the probability of the rank of true positives. And assume that the probability of ranks of true negatives are like this, right? Where you would put this. So that means that if the two were flat, this is a random prediction, right? The fact that I am assuming that there is some slope and some enrichment of good predictions here and enrichment of good predictions here for the negatives indicates that there is some predictive power, an epsilon, if you will. Now let's assume that the position where these things go is independent. That means that let's assume what is called a conditional independence given the class. That for team 1 I will sample the true positives from this distribution and put it here. For example, for team 2 I will sample independently from this distribution and put here. Suppose that we do this. That means that the positives are chosen in an independent way. If you do this, then it makes a lot of sense that the following will happen. I am aggregating the ranks and doing the average rank from this distribution. That means that I will be converging to the average of this distribution, which is here. And the negatives will be converging to the average of this distribution because I'm doing the average rank, right? Now, if I aggregate 5 methods, it might look like this. If I aggregate 30 methods that have some amount of predictability, then the variance will shrink towards the mean. And so I am assuming that all have the same mean. Now, so we can relax this assumption and still the thing works, but you understand what's happening? All the ones that are true positives, and I am doing the average rank, will, because they are independent, will tend to its mean. I am aggregating from a distribution that has a mean, and it will tend to that mean, and the variance will shrink as 1 of the square root of the number of things that I am aggregating. So in this case, what happens, you can, this distribution, the probability of positives having a rank R, you can write it, the probability of negative, you can rank it. We use the border aggregation, which is the average of the ranks for each item, and each item is independent, so you can estimate the 95% of positive and negative interactions. So you can see this is the average, and as k, the number of things that aggregate, this is the average, sorry. As k increases, the variance decreases 1 over the square root of t. This is for these distributions. And obviously, as k becomes bigger and bigger, this is going to segregate. These two distributions will be disjoint. And so you have perfect prediction, because if you are going to do area under the rock, you are going to first have all the positives and then all the negatives, and that gives you area under the rock of 1. So basically, that is, I think that's it. That is why this gives us some hint as to what are the assumptions needed for the wisdom of crowds to work. One is you have to have some predictability. Even an epsilon will do. The second is you have to have predictions that are independent, conditionally independent given the class. If that happens, then the more you aggregate, the better you will be. And that's what we have observed systematically. Now, oftentimes, we are not under those assumptions, so the actual aggregation is not better than the best and doesn't asymptote. But in general, you have seen that as we aggregate more, the median goes increasing.
So I'm largely recapping some of the things we've already seen, but very briefly. So what we learned from the real challenge, right? As we discussed, the challenge was about finding causal links, so network inference, in particular causal links, apply to human cancer. And as you remember, we had two versions. The in silico one that, as Pablo disclosed, was based on a real model or on a model that reflects real pathways in breast cancer cell lines around the RBB family and the downstream MAP kinase and PI3K pathways. And then from here, what we had done actually is to map some of the so anonymize the ligands. There were two growth factors, EGF and heregulin 2, what we call LIG1 and LIG2, and three drugs, or precisely one drug and two antibodies that bind to two receptors. And we had also mapped basically the proteins to the antibody, including some of these dummies antibodies, which were, as also Pablo explained, not in the real network. We were a bit to make things more confusing for participants. But then what we could do, if you want later, is to really compare your networks with the real network and with the real proteins and see how well it reflects the results. But what I wanted to do most over just 5 minutes or so is to show you a bit more on the real data. Okay, so the real data is quite similar to the one you use, but it comes from actual experiments that in principle tries to look at the same type of data, same type of system. These were 4 breast cancer cell lines, and these are used often as quite representative cell lines of breast cancer, and was data collected in Joe Gray's lab. And then what they had done is to treat them with stimuli and inhibitors and measure phosphoproteins. So it's the same layout as you look in silico, slightly different numbers, like number of time points of readouts and so forth, but it's really the same layout. And the same way as we have discussed around the in silico, we did the same type of questions. Can you infer the network? Can you infer the trajectory? And the scoring was the same ones, RUC curve or RMSE. So Pablo already showed you the in silico results. And again, and as Gustavo said, this is a common thing across challenges. There was really no clear relationship between the type of approach used and the results, or even if you try to compare in silico and real, even there it changes, right? So it's really quite subtle and not quite clear what is the best method. But what we can do is we can do meta-analysis of their submissions and try to learn things that seem to hold generally. For example, one thing we could do with the real data is to look at the value of using prior knowledge. Not in the case of in silico because it was anonymized, but in the real data, we had knowledge about specific proteins, and you could go to a database like Pathway Commons or other places to find what is known about those pathways and build into your models. And when we analyze within the models and compare how well did those that use the prior knowledge and those that didn't, you can see that it gives you a boost. So the pathways in databases are not perfect, but they give you useful information. And this is shown here, and this graph on the right shows you more or less the same information where we compare how well team did on the experimental and on the in silico case, and the ones in blue, which are the ones that use the prior knowledge. You can see how they get a boost in experimental data, which obviously not in the in silico because there was no prior knowledge. So as in the other cases, we found that the wisdom of crows holds, and this is the same type of analysis that Gustavo explained of aggregating submissions randomly, and as in that case, as you aggregate more and more teams, you get a better mean or median and also smaller standard deviation. And again, this holds both for the in vitro and the in silico data in a very similar fashion. To give you a flavor of the type of results, this is one of the examples of networks. This is an aggregate solution, so we're really combining all the different methods, and it gives you a sense of the level of wiring that we found in the real data, and then also how we were scoring. So one thing that also Pablo explained is how we try to find how many of these causal links or causal descendants are present. And as you can see, for example, here, right? So there are these chains of proteins in a cascade. They are all descendants of the same one, so they're all connected in how they respond to a treatment. And this was reflected in the scoring and in the results of the models. And another. So if you perturb something upstream, so you should see that the effect happens as a cascade.
So basically, you were perturbing either the receptor or.
Yeah, but for scoring, we use the perturbations with one drug, with mTOR, that we withhold, and then you should see that here. So when you block mTOR, you should see this one affected, this one, and this one, this one, this one, this one. But the blue ones are the false positives. So there are. Because this link is not there, all of these that were called as descendants, they were in truth not. So that's how you were scoring like this. So an interesting insight that you can do once you have these models and you can analyze them, right? At the end, what we have is we have network inferred for different cell lines under different conditions. The conditions were this stimuli, that means different growth conditions. So you could think of different cancer cells in different environments, exposed to different stimulation. And one question that we ask is, how different are all these networks? And what is more important, like the cell type, so the genomic background of the cell line, or the stimulus, or the environment where they are exposed. And we can look at the differences at the network level, and if you just try to look at the similarities in a simple PCA plot, you see how the genomic background is more relevant than the stimulus. So they are more similar. Those that have come from the same cell line, so the same genomic background, are more similar than those. So all of these cell lines, or all of these networks, are the same cell line under different conditions, and they are quite similar to each other, and the same holds for the others. So it's a kind of interesting insight. And also I wanted to mention briefly, yes?
So on false positives, for these cell lines, you have positive validation that those indeed are false.
Yeah, because we have data. So these cell lines are perturbed with different drugs and so forth, and we kept the data using mTOR inhibitors, and that's what we score against. So we know experimentally, when you put an mTOR inhibitor, which genes are going to change, and then that gives us the true or false positives, and that's what we score against. And yeah, and just to give you a flavor of the best performing methods, also as was discussed before, probably the most important thing is the combination of methods, but still there is certainly value and merit in performing best. The interesting thing was that two methods, different methods from different groups, were the best performer in the experimental data and in silico data. And the best performer in experimental data, which was the Stuart Lab in UC Santa Cruz. So on one side, they spent quite some time building a very good prior knowledge network from Pathway Commons, and they did different integration strategies to find the best subnetworks. And then they combined this prior knowledge with a method based on Granger causality that, if you haven't heard about it, is a way to see if basically there is between time series, if there is a causality in the sense that you can predict one from the other. And actually they expanded these concepts in a number of ways. Actually they call it the prophetic Granger because it uses also future time points to predict a given time point.
But then the best performing in silico data, and actually if you combine both things together, it was the most consistent one. It's a different one, very different basis that come from Jo Song's lab, and it's called Funshik. So this method did not use any prior knowledge, which also perhaps explains why it didn't so well with experimental data. And they discretize the data, and then they basically they develop a method. So there is there's the known person chi-square test to see if two values, if the distribution of values are different or not, and they develop a variant or improvement of that they call the functional chi-square test. And they use it to find relationships, causal relationships between the different variables, and they did very well on the challenge. And so these methods are also available in CINI if you have to download them further, and also they are described in the paper. Because that's I think one thing which is really nice, that when you run these challenges, you get a very nice resource in terms of methods. So I'm showing you here, this is the supplementary materials of the manuscript that is under revision about this challenge. And then you get, for all of these methods, you get this really nice description. This is actually the Granger causality. It explains you how they do this, right? So then you can go and look, what type of methods people try. You can get ideas, you can think of combining things, and so forth. But this type of resource value we will discuss a bit more later in the session.
Do you think it would be interested to embed some sort of annotation so that people could develop methods that produce prior knowledge, but then that would be applicable to the experimental?
So what do you mean by annotation? Because in silico it's all anonymized, right?
Yeah, but to try to enrich in silico with similar kind of information that you would find in the experimental.
Like basically using real proteins or giving potential information about what is the prior, what is the potential links in the proteins. Yeah, it's a good idea. I mean, we discussed, of course, because we could have given, we didn't have to anonymize the data. Our fear was that people could find the actual model because it's a relatively well-known model on the field. That's true. And give pseudo prior knowledge. Yeah, that's a good idea to explore in a control setup the role of priors. And yeah, actually, it's a good idea. But yeah, we didn't. Yeah. But yeah, that's all I wanted, just to give you a flavor of the few things we learned on running the challenge about, the prior helps, the prior doesn't help, analyzing of networks, and some of the methods, and one more time showing all the people that made this challenge possible. And unless there are other questions about this specific challenge, we can just show you briefly the tools that we use for the scoring, these dream tools. I don't know, Brian, if you want to show that here. Shall I open just GitHub or what is the best place? The GitHub? So we've been recently, and this is in particular Thomas Coquelin in our group at TVI, trying to bring together all the different scores from different challenges. Because as you have seen, right, so for example, even in this challenge there were a few methodologies, but in different challenges there are others, and it's useful to have access to this because first, maybe as a researcher, you want to develop your own method and you want to analyze it, or you want to do your own wisdom of crowds studies. This is also blocks of code that then can be reused by us in the signups to run the challenges. As new challenges will probably look similar in some ways to previous challenges. So we're really putting this together into here. And what you can do, if you are interested, is to go and use it and so forth. What is really nice is so you can run it with very little code. So you really, I mean, once you download it and so forth, you can simply, with one line, you can score any submission to any challenge. Yeah, you say you develop a method for this, and you can just type Dream Tools scoring, the challenge name, the sub-challenge, and say which file you want to score, and it will automatically, either it has the golden standard in the package if it's small, if it's large, it will fetch it from Synapse, and then you can get your scores. And here you have a list of all the ones that we have included. It's not all of them, but we keep adding them, and we hope to include most of them. As of now, it's like 2/3 of the challenges that we have the code here. And yeah, for we can even show you one of these Python notebooks applied to the challenge that you look at, and we can see how you generate the wisdom of crowds plots. And this is it through this link, Brian? Do you know? How?
I think that's the Synapse page,
Okay.
But,
It's in here.
you want me to drive?
Yeah, sure.
So if you drill into the folders here, Dream Tools, this was Dream 8 challenge, and it's the first challenge. There's lots of documentation about how to actually, what the scoring algorithms actually were, how to run it. So there's nice documentation about each of the sub-challenges, how to get things going, how to set up your Synapse configuration so that if you have to pull anything from Synapse, that automatically creates the hook and logs you in, how to download some of the experimental data, which Julio was mentioning, and then information about the different types of scoring that's done. And I'm going to skip over some of this and show you, I think he actually has the Wisdom of Crowds aggregate AUC here. So this is the type of plot that gets output from the code that he's generated. So I've noticed he's been working on it again recently. It's continually growing, like Julio said. There are a lot of the challenges are now populated within the Dream Tools framework. And actually, as promised, I did the Wisdom of Crowds for our mini challenge here. So if you go back to the workshop page, it's not as pretty, and I'll try and make it a little bit prettier over time, but I only had while Julio was talking here. Oh, the leaderboard first? So I don't have this ordered by winner, but as you can see, each one of the two teams did get a submission in here, and nobody was below random. So,
Well done, guys.
So that's good.
It's not so, I mean, people spend months of time below random.
It's true. And so I just threw another wiki page in here with the aggregate scores, so you can see individually this is what each one of the scores look like, and then the ensemble you can see always has done better.
So you four are together are pretty competitive, huh?
Yeah.
Think of what actually what the best was. We can go to. Oh, do you want to show it?
The best was the one that.
No, I mean the real participants, right?
It was actually the one that you sent on here, and I don't know which one that was. Yeah.
No, but if you see, for example, the aggregate of U4, right? After some time, it was already. I mean, compared to if we now look to the people that truly participated.
That's fine. Are you aggregating twice yours, right?
What's that?
There's two submissions from you.
Yes.
But they're different ones.
Sure. One Julio did and one you did.
Yeah. So this was the one that
different.
Julio did. Which one?
Mm.
And this is the one that I did.
Yeah, but what I think is interesting is just to give you, if you now go to the, I mean, I still have the slide.
I sent two submissions later after lunch.
I think they were invalid. Did you get emails? Okay.
Maybe it's the
They may not have.
Z problem. Or they say were valid.
If they said they were valid, I can take a look.
So
is it,
the same problem, remember?
is it
No, you said they were invalid. But,
No, I received an email saying they were valid. If you
okay.
receive an email, they're fine, right?
No. It would send it if it was valid or invalid.
Oh, okay.
So I mean, of course, one of the ones that we include in this ensemble that we submitted is one of the top performers, which was in this range. But thanks to your submissions, it got up a bit, right? And it got even, you would have gotten in. So if you guys would have participated, it would have been not bad. Where are we now?
Aggregate.
I was looking for the aggregate plots.
So it's, God. So they're aggregate all the way down.
Yeah. Okay. There you go. So you see how you boost with your submissions, right? It's what Gustavo was saying, even if it's...
Not to,
The first booster, the top two scores.
Yeah, the first two basically set the line, right? And then the others keep it constant.
Yeah. It's
Right?
true.
Fair enough.